Next is Roger Traub.
I'm going to talk about consciousness, not what it is, but some idea about how consciousness can be initiated by sensory stimulation. So, let's see, this tall guy, not the nerdy fellow on the left, but the tall one was Miles Whittington. This picture was taken in Mallorca. I worked with him for almost 30 years, and he died last October of leukemia. And so this talk is going to be a very quick summary of two of the last papers we wrote together. So the first one is going to be some insight into how the piriform cortex or olfactory cortex deals with sensory inputs, and then a hypothesis which was just published in PNAS about how sensory inputs through the olfactory system may or may not evoke conscious awareness. So a super quick sketch of some of the anatomy of the olfactory cortex, which I'm having trouble seeing. This is a phylogenetically very old three-layered cortex, vaguely like the hippocampus. It has pyramidal cells in its layer 2 and 3. The afferents from the olfactory bulb come in here, and the LOT, or lateral olfactory tract, make synapses on the distal dendrites of the pyramidal cells. And this system is extremely rich in excitatory recurrent collaterals, which is a very long-standing interest of mine, which came through my actually meeting Bob Wong at Stanford in 1977. And there'll be more of that in a second. So this is a sort of summary of the behavior of the system. This is in simulations. I don't have time to show the experiment. So what you're looking at is where this blue arrow is, the afferents start coming in. It's sort of a random splodge of spikes in these afferent axons. These are raster plots of some of the different cell populations in this model olfactory cortex. And these three simulations are at different values of a parameter which the AI people would call synaptic weights, or the strength of the recurrent excitatory synapses. And what you see is that as you increase the strength, the same random splodgy stuff coming into the system organizes to the point where you can recognize obvious, what we call cell assemblies. Here, this is a population of interneurons in pyramidal cells, and it shows up as a small field. So there are two things that are interesting here. One is this is a property of the system which allows it to become epileptic. If you overdo this parameter, instead of acting like an associative memory, which is how people describe the system, the associations start to make more associations. And you get what's called a phase transition by statistical physics people, and the system kind of blows up. But the point of it is, for me, is that this system of recurrent collaterals is actually what allows the circuit to perform a useful function, provided it doesn't try to overdo it. And you'll see that same thing in the next part of the talk on another part of the system. Now, the basic physical idea came from two people in the audience, both of whom have gray or white hair, and 40 years ago published this figure that's on the right. So the key experimental observation made by Bob was that in a disinhibited slice of CA3 hippocampus, a very small stimulus would evoke a synchronized burst like an interictal epileptic spike, but it would only do so after a latent period of the order of 100 milliseconds. And the question was, how do you explain this latent period? And the answer turns out to be simple once you see it, and was experimentally verified. You want to have enough recurrent connections, but not too many, and each of the recurrent connections should be sufficiently powerful. So this turns out to be a rather general phenomenon, and something similar is observed in slices of the piriform cortex with its underlying structure called the endopiriform nucleus, which is this. Karen Gale called this the area tempesta. It's one of the most epileptogenic regions in the entire brain, and it lies underneath the piriform cortex and is densely interconnected with it. Now, this is an example of an in vitro kindling, not like in vivo, like you heard before, from the lab of Lou Haberle. He would bathe slices in low magnesium. They would develop synchronized discharges. He'd return to normal medium, and then you would see the same kind of phenomenon that Bob saw in the hippocampal slice, namely a small stimulus would evoke a synchronized burst with a delay of the order of 100 milliseconds. Now, what does this have to do with consciousness? So Miles Whittington pointed out to me that an odor presented to a human might be perceived or not perceived, and there's a MEG or magnetoencephalographic signature. If it's not perceived, there's a rather weak localized signal. If it is perceived, there's a much more general signal over a large part of the brain, and again with a delay of hundreds of milliseconds. So the hypothesis that we proposed was that the endopiriform nucleus was exhibiting behavior very similar to what Bob had shown in the disinhibited hippocampal slice. In this case, inhibition is present, but we know from Lou Haberle's work that this kind of phenomenon can still occur with inhibition present if the parameters are chosen correctly. Now, how to choose them correctly, I have to refer you to the papers in this very short talk. So the result is that depending, again, on choice of parameters, an identical olfactory stimulus as here in a non-unconscious situation would evoke some recurrent activity in the piriform cortex, not involve the endopiriform nucleus, which is part of the claustrum, and which is hypothesized by Francis Crick and Christoph Koch to initiate conscious perception. It doesn't go anywhere. It sort of stays in the piriform cortex, can be forwarded to lateral olfactory, amygdala, entorhinal cortex, but doesn't involve the claustrum. A small change in parameters evokes instead a delayed synchronized discharge in the endopiriform nucleus and in the piriform cortex, and this is the sort of nucleus of our hypothesis of how conscious perception of this smell occurs. So, thank you.